Okay.
Okay, so the first slide, information slide is the top causes of death, and he really stressed in the lecture that you need to know the top 3 causes in the United States and in the world. So it's all the same like now. It's heart disease, cancer, and stroke. It varies, like the cancer and stroke is like inverted with the United States and the world, but that's what it is now. As compared to before, there was a fight for fighting infectious diseases like pneumonia, tuberculosis, etc. So those used to be really big problems, but now, because of antibiotics and water treatment and everything, the top 3 causes are heart disease, cancer, and stroke. But in developing countries, bacterial infections are still an issue. So, but yeah, 100 years ago, there were mainly infectious diseases that caused death. And he mentioned that the first fight against infectious disease from the Department of Health was water treatment, like chlorinating the water to kill bacteria. The second one was antibiotics, the development of penicillin and whatnot. And the third one was vaccination. So those are the 3 important reasons I would think you should be aware of. So yeah, remember the top 3, and also remember that around the world there are a lot of places that lack clean water and sanitation, which is what causes these infectious diseases. Yeah, okay. So the next slide is about the milestones in microbiology. And he said to remember a lot of scientists. So they included Antony von Leeuwenhoek. Sorry, I'm really bad at like pronunciation and stuff, so if you want, you can correct me. He pretty much invented the magnifier because he worked in textiles, and he was able to observe the first microbes on fiber. Remember Edward Jenner, who developed the smallpox vaccine because he noticed that milkmaids, because he realized that the milkmaids were not getting sick because they had been inoculated with cowpox since they worked with cows a lot, etc. Remember Semmelweis, who advocated hand washing. John Snow, who was an epidemiologist that studied cholera. Remember Louis Pasteur, who was the father of microbiology, and he's the one that did the swan neck bottle experiment. Joseph Lister, who had the aseptic technique of not contaminating anything. Robert Koch, who had the postulates, which we haven't really talked about. We'll address them later, though. Remember that he worked with anthrax, pure cultures, and he was the one that came up with the whole 1 microbe, 1 disease thing. Remember Alexander Fleming, who discovered penicillin. Paul Ehrlich, who found that salvarsan, he developed salvarsan, which cures syphilis. And he was looking for the magic bullet to cure, like a cure-all that kills any infection without having an impact on the body, which we now know isn't really possible. So yeah, for the next slide. Okay. This slide pretty much just tells you the different areas of microbiology. Just like remember some of them, the names like industrial microbiology, environmental, etc. And just know that it's a very diverse field. You probably won't get that many questions on this, just like kind of know it, like be aware of it. Okay, so the important stuff from this slide is the definition of a microbe. Is there anyone that can tell me the definition of a microbe? Yeah?
Can't be seen with the naked eye.
Okay, yeah. It can't be seen with the naked eye, so you need a microscope to view it. The 4 microorganisms that he talked about were bacteria, fungi, parasites, and also viruses in parentheses, though, because they are acellular. And all microbes are made of cell, and because viruses are acellular, technically they don't count, but still, yeah. So remember that a cell is a unit of life, and remember the hallmarks of a cell. Is there anyone that can tell me some of the hallmarks at all?
Replication.
Sorry?
Replication.
Okay, replication,
Cell membrane.
cell membrane. Anything else?
Independently.
Okay, same thing as replication, but yeah.
Controlling what goes in and out.
Okay, yeah, so regulation of nutrients across the membrane. What's the 4th one?
Uptake of nutrients.
DNA.
DNA. Yeah, so they have to have DNA. And because viruses are acellular and some of them have RNA, even though some of them have DNAs, technically they are, like, they're still microbes, but like Dr. Ashari says, like, put them in parentheses. Yeah, and other things that you need to remember are prions, which are also acellular, they cause mad cow disease, and viroids, which are in plants. Alright, so the next slide pretty much just talks about what microbes do. They were the first living creatures on the earth, and they prepared the world, like the earth, for for greater organisms. And pretty much they have the power to transform themselves or their environment to be able to survive in it. So you will have extremophiles like archaea that live in really extreme environments. They can change their own pH inside, or they can change their pH outside to be able to survive. They're more abundant and more diverse than any other living thing. They grow in every ecological niche on the earth that has a source of liquid H2O, which is a really important point because you really stressed that in the lectures. They participate in symbiotic relationships. They can recycle from inorganic to organic compounds back and forth, and only a very small fraction of them cause diseases. And so, also we learned a lot about genetics from microbes by studying them, and because they're so cheap and they're easily usable in everyday labs and stuff. So our basic knowledge of microbiology and biochemistry are from microbes. Are there any questions so far? Okay, good. Okay, the next slide covers prokaryotes and eukaryotes, the major differences between them. Who can tell me some of the characteristics of a prokaryote that's compare and contrast with eukaryote? Flagella, okay.
Eukaryotes have them too, though.
So, yeah. Sorry?
Peptidic glycan layer.
Okay, yeah, so that's only in bacteria so bacteria are prokaryotes, but there's also archaea that are prokaryotes. So you need to remember that only bacteria have peptidoglycan, which is targeted by penicillin and other antibiotics, like the cell wall structure. Eukaryotes do not have peptidoglycan in their cell walls. Some of them have cell walls, some of them do not. Prokaryotes do not have membrane-bound organelles. They also have only a nuclear region. And also, he really stressed remembering that prokaryotes have plasmids, which are extra chromosomal pieces that just float around. And they're really important because they carry genes that are infectious, that can transmit infectious diseases, or they can have antibiotic-resistant genes. So some of these plasmids end up being really important, and not only are they passed on from vertical gene transfer, but it's also horizontal gene transfer, so it doesn't have to be from parent to offspring. It can be between bacteria. Okay, and viruses are neither prokaryotes nor eukaryotes because they're acellular. And also remember some of the shapes, like the split shape, the cocci shape, the vibrio we talked about, and the spiral ones. He probably won't ask you that many questions on it, but just again, be mindful of that they exist. So again, there are 3 domains of microorganisms: bacteria, archaea, and small eukarya. Viruses, once again, acellular. They're not in that same thing. And remember the size of them, which is it's in a later slide. It's in the next slide. So remember the size comparisons because you might get asked a question on that. But pretty much you just have to remember that eukarya are 10 times, 100 times bigger than bacteria and archaea. So that's, actually, yeah, it's on this slide, the sizes and stuff. So remember the comparisons of the sizes. Okay. So from this slide, pretty much what you want to know is that we all started from the same origin, and they developed into different the different domains of bacteria, archaea, and eukarya. And prokaryotes are the ones that made the planet again livable for everyone, and also, there have been studies done, obviously, that we extracted DNA and RNA from mitochondria and such and sequenced them. And by doing that, you can see the homology of the different related bacterium. And also comparing the RNA of different molecules, you can see the conserved regions that have not changed over time. So that also makes it easier to construct this tree of life. Yeah, so again, this has a size on it, like the size of the bacteria, archaea, and eukarya, remember that. And it's just like a summary, so just like if you read over it, you'll know it. And yeah, just look at this chart also because it summarizes what we just talked about. Okay. So you guys, this is like a really self-explanatory slide, you can just read it. Okay, so you guys need to know the bonds, like you guys have all seen the bonds before, like atoms, molecules, elements, everything. But just like in this course, just remember that like you'll have to know like which bonds are associated with which polymers and stuff, which we'll be discussing in like the next slides. So remember ionic bond, covalent bond, and hydrogen bonds. Like remember what they do, like ionic exchanges electrons while covalent shares. Hydrogen bonding, like it plays a role in the functionality and stability of structures mainly. So just like hydrogen bonds are generally really important. Other forces include hydrophobic interactions and the van der Waals force. And the van der Waals isn't exactly like it's literally just an interaction. But hydrophobic is also very important because it allows for stability in different molecules. Okay. All right, so you guys need to remember the 6 major bioelements, and also you really have to remember like the different elements that compose the proteins, nucleic acids, lipids, etc. So we'll go over them right now. So who can tell me what fatty acids are made of? Just fatty acids.
Carbon, hydrogen.
Okay, yeah, carbon and hydrogen. That was a question that got asked a lot in the chat rooms. People are getting confused, like if it's asking about the glycerol and the fatty acid or just a fatty acid. But if he asks you just a fatty acid, it's just carbon and hydrogen. What is the minimum structure for a polysaccharide? The elements. CHO. Yeah. So if you just remember CHO, that's polysaccharide. What about proteins? Okay, so proteins are carbon, hydrogen, oxygen, nitrogen, and also, in parentheses, sulfur. For nucleic acids, it's carbon, hydrogen, oxygen, phosphorus, and nitrogen. And who can tell me for lipids what it is? This is a glycerol and the fatty acids. Yeah?
Could you go over it one more time? Just a little slower?
Okay, yeah, I'll go over it slower. Okay, so for polysaccharides, it's carbon, hydrogen, and oxygen. For proteins, it's carbon, hydrogen, oxygen, nitrogen, and sulfur. For nucleic acids, it's carbon, hydrogen, oxygen, phosphorus, and nitrogen. For lipids, it's carbon, hydrogen, oxygen, phosphorus. And for fatty acids, once again, it's just CH. And also kind of know the other bioelements that because they play a role in enzymes. So you probably won't have to know it really well, but just like even for future reference, just know them. It'll help. All right, so you guys know all about ionic bonds and covalent bonds, I'm sure, and hydrogen bonds. Oh yeah, water is very important, as we all know, because its ability to regulate temperatures really well, and that's why it's like a good temperature buffer, and it's also, its hydrogen bonds also helps its ability to be a really good solvent, and also because it's polar, it can expand. So water is a really good solvent and can regulate temperature because it takes so long to heat it up and also to cool it down. So yeah, just know those things about water. Okay, chemical reactions. Mainly, I would say, remember like that catalysts are what like makes a reaction possible, but catalysts are not consumed in the reaction itself, so they're always regenerated. And enzymes are biological catalysts, so they'll never like get used up in the reaction. They're just kind of there to like lower the activation energy. And remember that exergonic reactions are spontaneous and that like no push is needed. But for endergonic reactions, you have to like fuel it with energy for it to like go. Okay. So some of the chemical reactions you'll have to know, especially this one, is the dehydration synthesis and also hydrolysis. So dehydration synthesis is when you take out of water, and that's when you're forming a bond because you're taking out like a group so that like the two groups can like form together. Shows on the slide up here. And then hydrolysis is when you add water to lice to